Welcome everyone. In this lecture, we will discuss part 3 of this chapter, Biosignaling. Here, we will examine receptor tyrosine kinases, receptor guanylyl cyclases, and multivalent adaptive proteins. Receptor tyrosine kinases are enzyme-linked membrane receptors. Many membrane receptors consist of an extracellular ligand binding domain and an intracellular catalytic domain. For example, receptor tyrosine kinases have an extracellular ligand binding domain on the exterior of the cell. And this particular extracellular ligand binding domain is bound by a ligand which activates a receptor. The activation of the receptor activates the intracellular catalytic domain. The catalytic domain has enzymatic activity. The most common catalytic domains have tyrosine kinase activity, which means that the enzyme can phosphorylate tyrosine residues on its substrate. To begin with, the enzyme adds a phosphate group to itself. The autophosphorylation, as it is called, leads to a conformational change allowing binding and catalytic phosphorylation of specific target proteins. So the enzyme needs to be autophosphorylated first so that it can be activated. It further adds a phosphate group to tyrosine residue in specific target proteins that are the substrate of this enzyme. And as a result, initiates a kinase cascade. Insulin interacts with the cell via a receptor tyrosine kinase. Insulin, as you may know, is a peptide hormone that is produced by the beta cells of islets of Langerhans in the pancreas. Insulin reaches target cells such as liver, muscle, or fat tissue cells via bloodstream. Binding of insulin to the insulin receptor initiates a cascade of events that leads to increased glucose uptake and metabolism. The inability to make or sense insulin can lead to diabetes, specifically type 2 diabetes. Insulin signaling cascade begins with insulin binding to the insulin binding receptor protein. The active insulin receptor protein consists of two identical alpha subunits protruding from the outer face of the plasma membrane. Now the two alpha subunits are shown in pink color and in a brown color. Now these two alpha subunits also are connected via a transmembrane helix to two different beta subunits that are found in the interior of the cell or on the cytoplasmic side. Now the alpha subunits contain the insulin binding domain as shown here. Insulin is this red colored molecule that bind on the interface of the two alpha subunits. Now once the insulin molecule binds, a signal is transmitted through to the beta subunit, and the beta subunits get activated. How does these beta subunits get activated? It so happens that one of the beta subunits get phosphorylated by the other beta subunit, and it is the beta subunits that has the enzymatic activity. Now shown here is one of the beta subunits that is inactive and unphosphorylated. In the unphosphorylated stage, an aspartate residue interacts with a tyrosine residue, and in this case tyrosine 1162, and as a result of this, the activation loop that is shown here blocks substrate binding site. So it's in an inactive conformation. As a result of phosphorylation of these tyrosine residues, as you can see, there is a conformational change which moves the activation loop away from the substrate binding site. So phosphorylation activates by moving the activation loop away from the substrate binding site. Now this is the substrate binding site, and as you can see, a target protein binds in that site. And this is the target protein that gets further phosphorylated. So the takeaway from here is that autophosphorylation activates this tyrosine kinase receptor, and as a result it can phosphorylate further proteins downstream. So this is just the beginning of the signaling cascade initiated by insulin. The insulin signaling cascade is further amplified when IRS1 or insulin receptor substrate 1 protein gets phosphorylated by receptor tyrosine kinase. Now as you can see, once the receptor tyrosine kinase gets autophosphorylated, it is completely active and it can bind a substrate and phosphorylate it. And that's what happens to IRS1. Now once IRS1 gets phosphorylated, it binds to another protein called GRB2 or growth factor receptor bound protein 2. Now the way IRS1 interacts with GRB2 is via an SH2 domain. SH2 stands for SRC homology 2 domain, and GRB2 has this SH2 domain which interacts with a phosphate on IRS1. GRB2 does not have any enzymatic activity. Its function is to recruit two different proteins. One of them is IRS1 and another is SOS. SOS is another adapter protein, and the function of SOS is to interact with the protein named RAS. And at any point of time, RAS interacts with GDP and is in an inactive state. So as a result of GRB2 interacting with SOS, SOS interacts with RAS and replaces GDP with a GTP. And as a result, RAS protein gets activated. Now, what is this RAS protein? This RAS protein is a prototype of a family of small G proteins that mediate a wide variety of signal transductions. Now, if you remember the G trimeric protein that is involved in the G protein coupled receptor signaling, that has the GTP binding domain. The RAS protein is also similar to that G trimeric G proteins. RAS can exist either in GTP bound, and when GTP is bound, RAS is active, exactly like the trimeric G proteins. And when it is bound to GDP, it is inactive. RAS is a very small protein, and when GTP is bound, RAS can activate a protein kinase named RAF1. And so activated RAS binds and activates RAF1. RAF1 is a protein kinase, and what it does is it phosphorylates another protein called MEK or MEK. So RAF phosphorylates MEK at two different sites, and MEK is also a kinase which further phosphorylates another protein named ERK or ERK. ERK, when gets phosphorylated, gets taken up into the nucleus. And once it is inside the nucleus, it goes and phosphorylates transcription factors. Now, ERK specifically phosphorylates ELK1. And ELK1 phosphorylation is important because it modulates transcription of various different genes, specifically insulin related genes or insulin regulated genes in this case. And some of the proteins that are translated as a result of ELK1 activation are essential for cell division. So, insulin binding to the insulin receptor initiates a series of signaling cascades, and the end result is the initiation of transcription, which leads to the translation of various different proteins that are essential for cell division. And hence, insulin acts as a growth factor. Insulin signaling leads to glucose uptake and glycogen synthesis. How does this happen? The signaling pathway from insulin branches at the insulin receptor substrate 1 protein. GRB2 or growth receptor bound protein 2 is not the only protein that IRS1 interacts with. If you remember, IRS1 interacts with GRB2 via SH2 domain. The same way, IRS1, when it's phosphorylated, can interact with another protein named as phosphoinositide 3 kinase or PI3K via its SH2 domain. And once it interacts, PI3K is activated and it phosphorylates PIP2 or phosphatidylinositol 4,5-bisphosphate. PIP2, when gets phosphorylated, becomes PIP3 or phosphatidylinositol 3,4,5-triphosphate. PIP3 usually protrudes out of the membrane towards the cytosolic side. And PIP3 formation is the starting point for another cascade of protein kinases. When bound to PIP3, protein kinase B or PKB or AKT, it's another name, is phosphorylated and is activated by another protein kinase called PDK1, which is not shown here. The activated PKB has two functions. Remember this. The first function is to phosphorylate another protein named GSK3 or glycogen synthase kinase 3. When PKB is activated, it phosphorylates GSK3, and phosphorylated GSK3 is inactive. So it converts an active GSK3 to an inactive form. This is very important because GSK3, if not active, cannot phosphorylate glycogen synthase. And glycogen synthase, when phosphorylated, is inactive. But when GSK3 is not active, glycogen synthase now becomes active because it's not phosphorylated, right? Now when glycogen synthase is active, it can synthesize more and more glycogen. So glycogen synthesis is accelerated. The second function of PKB, as shown here, is to stimulate movement of glucose transporter GLUT4. GLUT4 or GLUT4 is moved to the cell membrane via vesicles, and this is to increase uptake of glucose. And so PKB activation results in uptake of glucose, and this glucose is then converted to glycogen by glycogen synthase. This is how insulin signaling results in glucose uptake and glycogen synthesis. The insulin receptor shown here is the prototype for several receptor enzymes with a similar structure and receptor tyrosine kinase activity. Shown here are different receptor tyrosine kinases. And INSR stands for insulin receptor. The receptors for EGF or EGFR and PDGF, for example, have structural and sequence similarities to INSR, and both have a protein tyrosine kinase activity, both these receptors. Many of these receptors dimerize after ligand binding. INSR is the exception, as it is already an alpha beta dimer, whereas some of the other receptors are not. For example, EGFR dimerizes only after the ligand binds to it. Extensive crosstalk among signaling system exists in cells. We have discussed the signaling pathways for insulin and epinephrine separately, but the fact remains that these two signaling pathways do not operate independently, and they are dependent of each other. Insulin opposes the metabolic effects of epinephrine. And what happens is the activation of insulin signaling pathway directly attenuates signaling through the beta adrenergic signaling system. For example, the tyrosine receptor kinase INSR for insulin directly phosphorylates two residues in the beta adrenergic receptor. So the other two residues are phosphorylated by PKB, which gets activated by IRS1, which is a substrate of the insulin receptor tyrosine kinase. Now, as a result of phosphorylation of the beta adrenergic receptor, we know that this results in the internalization of GPCR, right? And as a result of internalization of GPCR, the responsiveness of GPCR is reduced, and the concentration of this receptor on the cell surface is reduced, and as a result, it can no longer bind epinephrine. This is one mechanism. A second type of mechanism, or a second type of crosstalk between these receptors happens when one of the tyrosine residues is phosphorylated by the insulin receptor tyrosine kinase. And as a result of this particular phosphorylation of this tyrosine residue, this can interact with an SH2 domain of the GRB2 protein. The way this happens is by recruiting an SHC protein. Now, once GRB2 is activated, it further activates the SOS protein, which initiates the binding of GTP to RAS, leading to activation of RAF1, which is a kinase that phosphorylates MEK, and MEK further phosphorylates ERK, and ERK phosphorylation leads to phosphorylation of ELK1 in the nucleus. And ELK1 is a transcription factor, and we looked at how it initiates transcription of various different proteins that are responsible for cell cycle. And so these are the two crosstalks that exist. And so the fact remains that these systems do not function independently and are dependent of each other. Receptor guanylyl cyclases are receptor enzymes that, when activated, convert GTP to cyclic GMP, or guanosine 3,5 cyclic monophosphate. Many of the actions of this secondary messenger CGMP in animals are mediated by CGMP dependent protein kinase, also called as protein kinase G. Now, on activation by CGMP, protein kinase G phosphorylates serine and threonine in target proteins. The two types of guanylyl cyclases participate in signal transduction. One type is a homodimer with a single transmembrane spanning segment in each monomer, connecting the extracellular ligand binding domain and the intracellular guanylyl cyclase domain. So it's very similar to the insulin receptor in the sense that when the ligand binds, the intracellular subunit is activated and it has an enzymatic activity, and the activity is to cyclize GTP to CGMP. Now, the first case of receptors are the atrial natriuretic factor, or ANF, and this particular factor, ANF, is found in cells of the renal collecting ducts and vascular smooth muscle. The second set of receptors are called as the guanylin, or peptide hormone produced in the intestine with receptors in epithelial cells. Now, the guanylin receptor is also interesting because it is a target of bacterial endotoxin that triggers severe diarrhea. So both these receptors work in very similar fashion, and both have an intracellular GTP cyclizing domain. From our discussion so far, two generalizations have emerged. The first generalization is that protein kinases and phosphatases are important for signaling cascades, while kinases phosphorylate other proteins. Phosphatases remove phosphates from a protein or a set of proteins. Now, residues like tyrosine, threonine, and serine are the targets of kinases. And these residues are central to signaling, directly affecting the activities of a large number of protein substrates by phosphorylation or dephosphorylation. The second generalization is that protein-protein interactions brought about by the reversible phosphorylation of tyrosine, serine, or threonine residues in signaling proteins create docking sites for other proteins. Now, docking sites means the sites for other proteins to come and bind after recognizing it. And this brings about indirect effects on protein downstream signaling pathway. So many proteins in signaling pathway are multivalent, and they can interact with different proteins simultaneously to form multi-protein signaling complexes. Most of the proteins involved in the signaling at the plasma membrane have one or more protein or phospholipid binding domains. Now, as shown in this figure, few multivalent proteins are known to participate in the signaling. We saw that GRB2 have both SH2 and SH3 domains, right? SH2 domains to interact with the insulin receptor substrate protein and SH3 to interact with the SOS protein. So GRB2 can recruit two different proteins. The same way, there are kinases like SRC kinase, which have SH2 and SH3 domains. Phosphatases also have SH2 domains, as shown here. Now, many of the complexes include components with membrane binding domains as well. So, these particular domains are the recognition sites in the proteins that are shown here. And other proteins that have specific interactions with these domains will interact with these proteins to form multivalent complexes. So it is important to understand that the domains that are shown here are primarily responsible for protein-protein interactions in various signaling cascades.